Okay, welcome back. We will have a panel discussion with Professor Gaute Einevoll and the PIs from University of Helsinki and Aalto University who provided us the Ignite talks this morning. So welcome back, everyone. And I guess we're still missing a couple, but we can start with a question. What are the next big things, or let's have one thing. What's, in your opinion, what's the next big thing that needs to be answered in order for us to be able to explain how the brain works? So who wants to start?
If we knew what that thing would be, we would be working on that. So I think that's by definition, it's not something that that we would know.
If we think that there are several of those things, and there's one that you could solve, then what would it be?
Takashi, do you want to add something?
Yeah, so actually it's a bit difficult to answer that kind of question. But I mean, next big thing, it depends on the time scale, like next years or decades. It depends on that. But if we think about like years, I mean, for me, like more. For example, nowadays people are trying to, using organoid as a model of neurodevelopmental diseases or neurodevelopment or evolution. But so far, that brain organoid is not that great enough for that purpose. So we have to improve that, and people are working on that. But actually, now it's just a kind of brain-like tissue, but maybe we can make kind of mini brain-like stuff in the end, so that we can use as a model system, and that's kind of beneficial for everyone, I would say.
Yeah, thank you. And what do you think about the development of brain implants recently? The example with Neuralink. What do you think about that? Lauri?
How does it work? Sorry, I came a little bit late. Do you just do you raise our hand or something, or?
You can just talk.
You can just talk. Okay, sorry. Yes. No, I think this is, I was involved in, we've heard about this Neuralink and Elon Musk, which is sort of fascinating when you sort of also what happens when you have this engineering, this very forceful company style coming into neuroscience, and they have then made this cool, it's quite impressive, like little electrode, looks like a little coin with all this electrodes sticking into the brain, and of course it's nothing principally new. This has been done before, and I guess it's, we have cochlear implants that has worked as auditory, as I think it's 500,000 people in the world who can now have, as I understand it, quite good hearing because of this. But I think it's a long. I was involved in a project in the US where they tried to do the same thing and actually have a chip on top of the cortex to give blind people some kind of vision by sending electrical signals into the cortex. But I think it really this. I remember when I talked about it, people said, "Oh, then I can sort of get, then you can sort of maybe get see infrared directly via the electrode." And I said, "Well, sure, but why don't you buy night goggles, right?" I mean, it sort of is this. I think it takes. It will be very hard to beat the normal senses, well-functioning senses. So I think it's going to play a role for patients or people who lack one sense maybe. But there's, in addition to the technological thing, there's also obviously the the ethical things about starting to improve and doing surgery on healthy people to improve things. That's
May I try to answer the first and the second question at once, because I think they are related.
Sure.
Yeah. So as Eero said, if we knew the most burning question and the kind of an answer to that, so we're working on that. But the kind of thing that I think is important is to find yet another governing principle for the structure and function of the brain. I mean, we have the neuron doctrine, we have Hebbian learning, long-term potentiation, all kinds of things which seem to be ubiquitous in the brain. But still there are things like what Gaute was showing in his talk, that you can have different wirings, you can have different synaptic weights, but you still have the same overall behavior. And I think, so that's also a problem for these brain implants if you apply them to areas that are not, for example, the primary sensory regions, because in each individual, what is happening there has its sort of a individual characteristics. So then there would need to be a lot of adaptation of this kind of an implant to do something useful. Of course, we can do it with computers, but to actually learn something of brain functions. So I think we would need to find yet another governing principle that somehow sheds light on the fact that we can have so different wirings and still they implement the same function.
Yes.
I could add there that I think Lauri is completely right, and I think that we really don't have an proper theory about how the brain functions. I mean, what Hebbian theory and neuron doctrine actually are talking about individual cells and how they work, but a sort of theory about how these work together is something that we are still missing. And there are some now approaches towards that direction, and I think that this is something that should actually then guide the future development, so that if you do have a theory, then that should you start to test that theory and not just kind of continue ignoring that part. So one part which I think is very welcome is this what Gaute was talking about, is this working together with experimental work and theoretical work, and also then genetics can be fed in. So I think that the computation is something that has really marched into all areas of science, and that's something that and definitely into neuroscience. And brain is about dealing with information. So that's why I think that sort of computational methods are going to be ever more important especially in neuroscience.
I could maybe continue here what Eero said. I would actually also, or I think that understanding how brain works, we would first need to know what are the cells there. And we know and understand quite well the heterogeneity in neurons, and we have classified different neurons. And still we have, for microglia, we have two type microglia. We have astrocytes, we have two type astrocytes. And this is what is now happening, is that we start to understand the heterogeneity of these other cell types, which is still half of the cells in the brain. And before we know the different kind of microglia that there is, I think that it's a bit challenging to overall understand what's happening. So I would predict that next 5-10 years, what's going to happen is that we start to understand and rename the cells in the brain.
I can say, sorry, Riitta.
Sorry, go ahead. Yeah, no, I was just going to say, continue that coming from the kind of cognitive end. It's like, I totally agree with all the questions about all the approach of the need for modeling. I mean, that's clearly absolutely the thing we have to do. Also, if I think from the neuroimaging side, it's clear that we have come to a point where we kind of know what kind of effects we expect to see phenomenologically. We know what kind of responses we might be seeing in the brain, but we are still no closer to really understanding what they mean, what kind of computations are going on. And even here, I I'm talking of computations as a much more abstract level, at a higher level than what, I mean, I of course one has to start from the ones where you can, like, obviously visual system is the focus at focus right now, which makes total sense. But we all know that probably at the end, we've really like to understand, try to be able to use neuroscience to also understand how we as humans operate. Although, of course, we look at mice and are happy to understand mice better, but in the end, we would like to understand humans. And then we need to operate with these kind of really population level signals, which we can collect from functioning humans. And there, when I think of something like cognitive process, that's really very far away, far removed still from visual processes. And there are clearly models coming from that end, which are kind of abstract models of cognitive process, well, computational models, but they are, it will take a long time, I think, to really link them to these kind of bottom-up models. But we need to be there. We need to. I don't think we can really move forward now unless we really start building quantitative models at all different levels and eventually trying to bring them together. But I think we are at that stage now, and that will be the next big step to really start to understand what we are measuring, now that we kind of trust what we are measuring.
Interesting, because I think you and I are probably on, like, in some sense, the opposite ends of terms of abstraction level, and I fully agree with you in the sense that we sort of, one thing we need this. I mean, at the moment we know that we are not too successful. I would say, depends on whether you think the glass is half full or half empty. But at that stage, one should really be. It's not clear what will bring the most success. Whether it's like this fMRI with machine. I mean, and we just have to try out different things and then be honest about it, and then try to also then communicate with all the neuroscience doing. I think some of this, it's when I came from semiconductor physics, is that which I used to do before I studied neuroscience, was that it was like, now some of the fights that I see within neuroscience, it's like, it's interesting that it seems more. That it's the less developed the field is, the more opinionated people are. It's sort of like it should be the other way around. So I think it's something about being a little bit, get this culture. We don't really know, and we should. But then you have to try. Of course, I use the physics approach because that's how I'm trained. That's how I can contribute. I can't do experiments. I can't do. I mean, that would be like I wouldn't be. I had to be retrained, right? So anyway, but from the physics point of view, this, when you come from this, like I bring with me the physics point of view, is that you, like, I think Feynman said that, well, if you understand it, you can build it. And for me, building it, meaning that you build a mathematical model that can mimic certain aspects from it. And I think what is the most limiting factor there is actually that at the moment, it's very difficult to deal with plasticity and all these homostatic things. I think parameters in the brain, like in the mouse, is set by, you start with, just like you do in a deep network, you start with a structure and then you train the parameters on the fly all the time to keep it, and we don't really, that's not typically not put into the models. And that's why we're struggling so hard to fine tune the parameters, because we have no plasticity that can sort of tune for us. So I think that, I hope that when you get, and the problem with the computing of that is that if we typically do a simulation, we simulate maybe like 1 second, because that's the response to some kind of stimulus. But if you want to have like this plastic processes in, then it's like hours, days, right? Often that, and so it's in order to run models for so long time that you can test out these plastic things is required computer power. But I think plasticity from my niche, my aspect of neuroscience, then I think plasticity, to get that into the models, I think that will help us actually. It will be easier to tune the models when we get the plasticity in.
We have one comment for Mikko, I think. If we believe that cells do computation as the most important function, do you really think that revealing different cell type will advance us much on how the brain works? Oh, you are muted.
I'm not really sure can I answer this question, but this is basic principle. So if you take any neuroscience book, these are our Bibles. They don't even mention microglia or astrocytes. They might mention oligodendrocytes. So how we have this neuron-centric view of our field, and we are completely missing. Then we also know that microglia is essential for synaptic pruning. Without astrocytes, there wouldn't be any activity in neurons. So and if you think about development, all the neurons have radial glial origin, and it doesn't go away. So actually, probably most of the transcripts, this is now my opinion, and it's not a proven fact, but I bet that all the transcripts that are found in astrocytes can also be found at some level in neurons. So just to keep something in mind, but I'm not really sure, can I answer that question?
I just want to say that I fully agree. And then actually, last year we published a book. I was involved in the book, and also Maria-Lena Linne, on computational glia science. So even though it's completely true that glia has sort of been neglected, certainly by the computational field, it's sort of now, coming back in. It's been taken up. So it's also work along those lines.
And maybe I can just, if you think about the lymphatic system, I think that is probably one of the biggest discoveries from the recent years. So, and it kind of makes sense to clearing up the garbage from the brain while we are sleeping. This idea has been there for a long time.
There are many multidisciplinary groups here. So what do you think is the greatest obstacle for incorporating these different approaches in your work? For example, like using models if you're doing something completely different, if you're working with cells or working with cognition. How does one go about it, and what's the greatest obstacle there?
Well, I think the biggest obstacle is that it's the whole field is too sort of siloed. I mean that on different levels of measurement, you have your own models, and they don't really bridge to another level. So I think having models that are able or that are predictive at multiple levels, like different spatial scales, so those would be tremendously useful, but they are not really made, because to do those models in a proper manner, you would need to have expertise from all these levels, and it's complicated enough already on one level. So you would really need to bring these kind of people together and come up with such models. There is, of course, a theoretical and practical problem, that how do you simplify these models appropriately that they become tractable at higher levels? You probably don't need to have each neuron described by 1,000 differential equations when you are doing systems level neuroscience. But like, how do you take this step so that you preserve the essence, but then you still have a model that you can use?
I could say that I think if we think in terms of how to in practice do multidisciplinary work, so at least from my point of view, the biggest, actually a really big challenge is that you have to really get like halfway to the other discipline to be able to both ways. So you have to have enough overlap, and you only get that overlap by spending a lot of time in discussing and trying to define what the question is that you're actually working on. So I think in practice, how many disciplines you can kind of incorporate really truly in your work depends on how much time you actually have to spend in like becoming a part of an expert in that field as well. Not really an expert, but enough that you can really exchange ideas. So I think it's sometimes really it's a practical question of how efficiently you can make use of knowledge in another discipline, because you have to kind of be able to, and I think it's just always take a half a year, at least in my experience, when you try to incorporate a new field, a new discipline into your research, at least.
In physics, in physics, there's been like for 100 years, it's been like the division of labor between experimental people doing experiment and theory, right? But they have the same training. They go to certainly they study physics together, and they go also grad school partially together. So is this something, should we really have like one neuroscience program where you learn because I think that will certainly help the discussion later, right? When if they train some sense, have some basic training the same way, is that feasible?
Yeah, I think.
But also
I think
there
that,
are new.
I think,
sorry.
yeah, I think that partially what what Lauri was telling. I mean, we have these silos, and it's not only that we have them, but people in the silos actually have a culture and they have a language. And what Riitta was telling, I think it's partially. I mean, to become an expert, is big part of it is to understand the language. I mean, how the people are sort of looking at their data, and that may be surprisingly different, even though people are looking at the brain or neurons at the same time. But if you're using different sort of experimental systems and paradigms, you develop a completely different way of looking at the data and talking about it. And that I think is the and then maybe this is what Lauri was telling is that we develop a lot of details, but we don't see the forest out from the woods. And if we saw the forest out of woods, I think then the discussion between the disciplines would be much more straightforward, because you could talk about the forest and not about the woods. So I think that, but often, this is what often happens, is that you gather lots of details and you think that that's actually going to bring us forward. And in fact, this might actually bring and take you further away from understanding, the forest.
Iiris is doing a lot of genetic. Do you have some insight about the discussion?
Just about to say that I completely agree with Riitta that it takes a long time to get familiar with different fields. And, I'm a human geneticist by training, and then I did 2 postdocs in neuroscience. And now I finally feel that I'm able to do both genetic and neuroscience research. And somehow we're able to combine that. And I think one of the great things in this kind of research is that you can have young people in the lab from very different disciplines and have them work together. But it is much slower than if you're really concentrating on just one field. And I think the fear there is that you're spreading yourself too thin, that you're not really an expert in one thing or the other, but you're trying to do something in the interface. So I think it's really important then to focus your research questions to something that's meaningful and important.
Just say, because there was a very nice initiative started a few years ago to have these Nordic neuroscience meetings. But and I think I very much believe in Nordic collaborations. So I was on the board of Nordita, one of these Nordic research institutes, for many years, and I think we have many things in common in the north. And it also is 25 million people is a better unit for doing science than 5 million people. But I think I'm also a little bit disappointed about the Nordic neuroscience meeting, in that I think, like, for example, the computational neuroscience thing is like last year, I don't think there was anything. So it's not like a place that that me and my students would naturally go to. So I think, and so instead we go to like this, I think the Bernstein meetings in Germany, which I think is sort of like trying to be much more cross-disciplinary. So maybe that's, if we want this sort of like cross-disciplinarity in Nordic neuroscience, like there's one thing we could do is sort of trying to make this, like they say, the Nordic neuroscience meeting or some other meetings more appealing to all parts of neuroscience.
Yeah. Are you going to computational, Iiro?
Pardon?
Are you going a little bit toward computational neuroscience?
Yeah. So there was this discussion about, what are the big steps, and there were already, multiple very good answers, and I agree with the previous speakers. So maybe something specific, or a few specific ideas. One is, of course, like these advances in neuroimaging techniques, both in terms of what we can measure with, how good SNR and in how great detail, and also how do we analyze those data. So maybe these technological advances, of course, will pretty much determine what we can do. And actually, they're opening up possibilities to really study what is, let's say, unique for humans. How are humans as active agents in this world? Social cognition, for example, and also language. And then going to some specific idea that I'd love to look into. Those better methods available, such as columnar and a cortical layer specific imaging. So I would really love to look at what are the laws that govern distributed representations. We know that there are these sparse distributed representations that go with certain perceptual cognitive states. They occur at multiple hierarchical levels of the brain. They're very dynamic. So this also relates to plasticity. And that would be a very, very big challenge also from the point of view of multidisciplinary work, because then you need the computational side, you need the methods in terms of hardware and analysis algorithms, as well as really strong expertise in neuroscience, cognition, and things like that. Yeah, so this was sort of like the answer to all the questions once.
Thanks.
Yeah.
There's a question from audience. Shouldn't we all be working on brain scanners that would allow imaging of the whole brain at the single cell resolution?
Yeah, so if I could continue a little bit here, actually what 3 Tesla may offer, but not really in a great way, but 7 Tesla would offer is columnar and layer level accuracy, and this is actually there would be an important step towards this vision. So of course, if we were able to image all the cells, that would allow for many things, but naturally then there would be this challenge of how to make sense out of that. Yeah.
Okay, Takashi, what is your opinion on this whole brain imaging with single cell activity resolution?
Well, it's fascinating, yes, and well, it's again go back to kind of Mikko's comments. Like we need to do that. We need to understand what kind of cells inside the brain. Still we don't know that what kind of cells, how many of them, and this kind of brain map. It's not only connectome at the brain map, and of course the biggest kind of challenge is each individual has a different kind of types of the overall it's similar, but its details are different. So that kind of individuality, we have to think about the individuality, and also we have to think about like what part is a common, what is a part is individually specific. So that kind of a whole brain scanner, maybe we can. It's good to focus on the common part. Maybe it's not only not in the single cell level, but kind of single column or a tiny region. I don't know. And I'm not an expert of that. That kind of stuff. Maybe it's more beneficial for many people. That is kind of my opinion.
Thank you a lot.
We're approaching the end of the panel discussion. I have one more question that I would like you to answer as briefly as possible. How far away are we from actually understanding the brain? What do you think?
Maybe I could start and saying what does it mean to understand the brain? Because I think in terms of weather forecasting, it's a good example. We can compute now actually amazingly well, sort of like many days into the future, the weather. But it's quite unsatisfactory in the sense that, well, I think it's satisfactory if you plan a vacation, but it's unsatisfactory. It's like it's not it's not like any principle between like the basic laws of weather and between the weather forecast that you can sort of understand intuitively. So in some sense we understand it, and but we don't really have this idea that we should sort of like be able to understand it in terms of understands or like concepts that we could put into words. I think it's not going to be it's going to be better in the brain science, but it's also this question: what does it really mean to understand things? If you can build something that predicts things based on and you can also change things in there, like for example in the brain, change what's the action when you add some drugs, or like so that you can test whether a network is doing better, is healthy maybe. If you add some drugs, if you had that kind of, even if you don't understand like in detail, if you don't understand the principles in some sense, it still will be very useful.
Okay,
well, I think it's a moving target, so it's hard to say when it's just improving our understanding of brain function. But I think another thing which is useful for testing, not just that we can make predictions, but also that if we can understand the mechanisms of a brain disorder X, and then maybe even develop a treatment for that. So then this is of course an advancement, but these also go in steps.
Yeah.
Thank
And
you.
we also
very much
have
for-
mechanisms. Sorry, I was just going to say that we also understand the mechanisms at very different levels. That mechanisms also can be understood at many different levels, which we don't really. Yeah, but I agree.
Yeah, it's been a very interesting panel discussion. Thank you all for attending. We'll now have a lunch break, and then we'll have the next session, which is hosted by the Journal of
Reproducibility.
Reproducibility in Neuroscience.
Yeah,
So,
thank you a lot.